Okay, let's start for the first panel discussions. I'm very happy to introduce our moderator for this first panel discussion. It is Dr. Tanja Kostic, who is a senior research engineer at the Competence Unit Bioresources at the Technical University of Austria. No?
Austrian Institute of Technology.
Okay. Sorry. My mistake. Her research focus is on food system microbiomes. She has participated in various national and international projects and has extensive experience in research collaboration with industry and published more than 30 scientific publications, and currently she is coordinating the Horizon Europe project Microbe, dedicated to the development and of optimized methodologies and technologies to preserve and provide access to microbe samples and associated data to enable more efficient development of microbiome-based solutions and applications.
Thank you for this introduction, and thank you for giving me the honor to moderate the first panel. So we are now like a testing group. Let's hope that everything goes right. We have 5 panelists in the room, and we should have 1 online, if I'm correct. Yeah, great. So I will just like to start by asking yourselves to introduce yourself briefly with 1-2 sentences. We are 6 people, not so many time, and I will ask to start with Lene. Ladies first, and then we will make round in this direction. So,
my name is Lene Lange. I have been working in microbiome support action along with Tanja. And in short, what can I say? I have 20 years with Novozymes, Novo Nordisk, and here at Novo. And that was learning me an impressive amount of what it was needed for microbiome. And that was the first part of going into microbiome studies also. The next part, I've been professor at 3 different universities in Denmark and research director. I've done so many things. But the only thing I'm really interested in is microorganisms' interaction with their host and the substrate. And that is where I think it's impressive what you have been managed to do, what you have done in SIMBA project. I was only hearing this morning and I really enjoyed it so much. Thank you.
Yeah, okay. So I'm Alex Türber. I'm from Bielefeld University, bioinformatician by training and into microbiomes since like 15 years, I think.
I'm Søren Sørensen from University of Copenhagen. I'm a professor in microbiology here, and other than Lene, I've been at the same place always.
Daniel Neuhoff, University of Bonn, one of the minority agronomists in the SIMBA project, working in the field, not so much in the lab.
Anton Alberdi, associate professor here at the University of Copenhagen. I haven't been involved in SIMBA, but I was the scientific manager of the sister project Holofood. And I am also coordinating another H2020 project, which is called 3D Omics.
thank you. And we have online our FAO guest. So please, if you could introduce yourself.
Hello, good morning to you. My name is Selvaraja Ramasamy. I work with the Office of Innovation as a senior technical officer, focusing on research, agriculture research for development, and also extension and advisory services.
Thank you. Thank you all. So I think everyone here in the room, I will now assume that sees the potential that we have in microbiome research, in microbiome applications. And I would just like to give you an easy warm-up question. What are, in your opinions, the biggest remaining gaps and barriers we need to address in order to realize on this promise that we are seeing? So whoever wants to start on that one.
I'm ready to start to say I think we need to include all types of organisms. I was missing, I believe, much of the presented did not include fungi, didn't include protozoa and so on. And viruses, of course, have to be there. And if you don't find them, it's because you haven't the right data and the right analysis, because they are there. Then the other part I think is extremely important is regulatory. We need to make much of this in the knowledge will not be brought in use for benefit of the users of nature and human health and animal health and plant, because regulatory cannot follow and really understand it. So we need something on regulatory, we need to have all the organisms included. Otherwise, we cannot talk about understanding microbiomes.
Thank you, Lene. Anyone else who would like to? Perhaps from the data set, I find this always really...
Well, I have to defend myself a little bit, because when I get the data, it's already too late, right?
Yes.
It depends on the extraction protocols and so on, what kind of organisms you see at the end in the data. So I think that has to be planned in advance. And the gap I see in terms of the bioinformatics and the data management, and this is what I mentioned in the talk earlier, is definitely the metadata. So the data sets are mainly valuable for the people who generate the data. And although the data is available or made available in these repositories, metadata is a big gap. But I do see initiatives that are addressing the problem. So at least in Germany, there is a big initiative now, the National Research Data Infrastructure Initiative, which is funded by the German Research Council. And it's interesting to see that other disciplines, not the biology, not the life sciences, but other disciplines, they are actually way behind, I think, in terms of making data available in repositories, but still, I think even though the data is available, the metadata is a problem. And the big question is, we need standards for metadata, and how do you convince people that they actually submit their metadata when they submit the data to the repository?
Fully agree. Thank you.
Maybe a short amendment related to the practical relevance in farming in Europe, and there we see that these microbial applications, unless they are crop protection agents, so those we call biostimulants, we have the problem of the missing reproducibility of significant effects, and that would require more research to understand what are the reasons for this. I have some suggestions for this, but that's not the case to do that here in the panel.
Can I? Sorry. No, perhaps just following on what Alex was saying. What I feel we are missing is, I think, both culture and education on how to generate and how to archive metadata. And culture first, so that we prioritize that. Because it happens oftentimes that you run a project and then in the very last months or even weeks and say, okay, I need to archive all this data, and then you realize that it actually takes months to archive data properly. And the other one is also to have the education on how to create this metadata, following the standards, but also following ontology of the different metadata, because it's not the same to use a tax ID code or use a scientific name or the common name of a host species, for instance, so that then other bioinformaticians can say, okay, I want to retrieve all these plants, all these vertebrates, all this whatever, and it's much more interoperable in that sense. So I think these are two of the key things that we are still missing. It would be really nice that we start creating some education way before in this academic career, so probably in the bachelor time, etc., etc.
Well, if I should give just one final comment also on this, is that we actually here in Copenhagen in the spring had a meeting on bacterial genetics and ecology, and I think one of the take-home messages there was context matters. So I think that we have seen it in all the way down to genomics, that where a gene is in the genome is helping us a lot. We are developing the tools at that case to see what are the genetic context. But I think very much so for bacteria and microbes, the close microenvironment they are living in. So currently we are for most of these studies looking at the wrong scale to really understand it. So we are looking at 1 gram of a sample which has more bacteria than the whole world, and we're blending in and trying to ask who is interacting with who. And it's really not the scale to answer these questions. So I think there are scientific challenges there to get down, and because I think whatever is happening is a context-depending thing. So you need to know the context that the bacteria or the fungi are in the environment you want, in order really to understand this. And I think that's a scientific challenge for us for the next couple of years to really get to this level. But I think we're on the right track.
Mm-hmm. Thank you. Mr.?
Yeah. May I?
Yes, please.
Oh, thank you so much. Yeah, so just, from my side, the unpacking of the complex issues to promote field application of the microbiome research is very important. That is especially focusing on adding depth to our understanding of nutrition and how the diets and food products are mediated by the microbiome and what are the effects on human health. This is one of the aspects that we see that there is a gap. And addressing this gap, of course, this needs engagement of multiple stakeholders and bringing or bridging boundaries between science and policy or the core, and it can have a comparative advantage. For example, the science policy and the societal interface that is missing, especially linking to the research institutions and also the local consumers and also the producers. I'm focusing on the food production in this case, and how this can feed into the policy processes and also the prioritization of policy-related aspects focusing on this particular area of work is also there is a gap and there is a comparative advantage to promote this type of research. The next one is, we need to engage with the scientific community worldwide to identify and share innovative ideas and solutions that can bring rapid and tangible food systems change, especially the transformation of agri-food systems, taking into consideration of evolving risks and also the problems, including the climate change. The other area that I would like to stress here is, focusing on agriculture research for development. The investment is so low. For every $100 of agriculture GDP, the investment in agriculture research is less than $1, and this is about 0.72 cents on average. And when we focus on the least income countries, it's not even 0.5 cents. So it's so low. And some of the least and middle income countries, this investment has not grown since 1980s, focusing on agriculture research for development. So these are all, there's some high level major gaps that we need to focus on. And one of the areas that need to be stressed here is the science, policy and society interface. Thank you. Over to you.
Thank you. Lena, you wanted to add something? Mike, please.
And another thing I really think would be interesting focus, that is to try to include all the ones we normally don't see of the because we take only the full genome and then take the entire and then skip looking at the organismal level and try to look at what goes on between interaction secretome of all organisms in the microbiome. So it's both holistic, getting everything included, and it's reductionistic by saying what is interacting with host and substrate. And I think this is doable, and I'm myself working on the enzymes, the protein, where we have developed a method for it called conserve unique peptide patterns, and it works both on genome and metagenomes, but also combining with metabolites. We are silos, one silo on enzymes, another silo on metabolites. We need to combine those to understand the interaction between microbiome and host and substrate.
Thank you. So what I take from this round is that we need to be significantly more inclusive, significantly more systemic, contextual, as you say, and we also need to really go to this multi-actor approach and improve communication. Now coming back to this, let's call it a group of 5 projects that we were part of with SIMBA and the Master and the Holo Food and Circles and Microbiome support. A lot of effort was done over the past 2 years to address some of these issues. A lot of work was put into the standards, into creating metabases in different environments, into providing tools, into discussing with different stakeholders. Now at the end of these projects, I think my biggest question for everyone would be, how do you see the legacy? How do you see that we keep all of this good work and how we can use it in the future and not have a next round of projects starting again with, oh, we need an SOP or we need a pipeline or whatever. So how would you see, how could we as this community of 5 projects try to ensure the sustainability of what was done in terms of developments in the last 5-6 years?
Something that perhaps cannot be done, but something from the European Commission perspective could be foreseen would be the microbiome support project, for instance, any kind of support and project to be extended over the period of the 4 other projects that were actually doing science and generating data and generating also all kind of relevant information that should be shared across projects. So I think one strategic option would be, in hindsight, to do it the other way around. Not first the CSA to finish and then the other 4 projects to finish, but it would be really great. It would have been really great if microbiome support had lasted 2 more years, because that would give us the opportunity to gather all together and have these kind of discussions at a much larger scenario and with all the different partners involved properly.
Thanks. Microbiome support is continuing, but as an association. We tried that way, but it didn't work with the Commission. We suggested that.
So what we have heard this morning from you, Alex, is that the scientific machinery with respect to methodology is advancing with high speed. So I think scientifically there's a lot of hope that we get more understanding. But what I see is we need the link, not only interaction between the organisms, but also the interaction with the environment. And the environment in an agricultural field can be very diverse. And just remind, if you do a pot experiment, what you have to do is you have to keep the humidity always at 60% capacity to make a crop growing in this pot. But under real conditions in the field, you may have drought periods, you have flooding periods. And I think this, and also the temperature, which in a pot experiment is normally standardized at 20 degrees, while in the field, depending where you are in the world, you have a high variation and quite often under temperate climate, much lower temperatures. That should, from my point of view, be assessed systematically, but methodically it's not evident to really, for example, highlight the effect of temperature under field conditions. How do you want to change the soil temperature in a field experiment? It's very cost intensive. We need at least the Norwegian gas to do it.
I think actually what I heard this morning, you have in SIMBA project been really good at thinking about what can we do to make sure we empower and we make things available for next steps to be done so we can stand on each other's shoulders. That's what I heard. I heard it from you, I heard it from Søren, I heard it all over. And I think that is a very, very big credit for SIMBA as such. I'm really impressed. The other part I would like to say, this was the first time I really heard European data. Because normally we are, in genomes, like in fungi and bacteria, we hear everyone going to Genome Institute of Beijing, JGI, and it's all over. And then it is much that European scientists sending their sequence, and the sequences will be generated in US or in China or in UK, and it's not counting as European data. I was impressed to see how much we actually have European data, but it is not reflecting the amount of money we invest in it. We are still not that big. And I have tried many times, I'm in scientific committee advising the European Commission in the area of circular bio-based economy and BBI and so on, a lot of $1 billion euro all in all. And I'm trying to advocate for that European research support should be strengthened, because we don't have the same kind of level in European Bioinformatics Institute. It is almost invisible. I mean, it's not to offend them, but they have such a small budget compared to the other continents we are comparing us with. And I think it's important that we empower our and really build this much, much stronger. Please, all of us can advocate for it in different fora.
Yeah. Well, I think one of the problems is also the sustainability of the bioinformatics workflows, for instance, right? So we are developing these workflows, and most of the work is done by PhD students, right? And then the PhD students, they get their PhD, they go on to a different project, and then there is this software, this architecture, which has to be sustained. Otherwise, it's just gone, right? And I think there is funding missing to keep this sustainable. I mean, there is the EBI, of course. The EBI is developing their own workflows, but I would say they are more general kind of workflows, right? And you need, for specific questions, you need very specialized workflows, and these should also be made available in a sustainable fashion.
Yeah. We do? Okay. I think this is really what you say, the funding to keep up the tools. And also you mentioned something really interesting at the beginning, how to educate and motivate scientists to provide the metadata. Yeah? So as I said, I was also in the project where you said, okay, this is the metadata, SOP, what you need to do. But I think putting these into context and, even making sure that they live after the project is something that is key. And I think also that in Europe, we somehow miss this, let's call it meta infrastructure that would take up all of this good work and then, make sure that it lives on after the PhD student has become a postdoc somewhere else and after the project has ended. And I think we have similar issues also with the biological resources that are collected in the projects that we haven't addressed here. Being aware of the time, it's an interesting discussion and also something that our online panelists say. I would like to now address this issue of engaging and communicating with different stakeholders. And I would now start to see a perspective of a FAO, because you said a lot of this science policy interface. I think in the past few years, and these 5 projects were really challenged to do that, and we learned a lot. But how can we improve? Because speaking out of the shoes of a scientist, it's difficult to get in contact to a policy maker. So what would your recommendation to us be how to improve from our side?
Yeah, thank you. Thank you very much. This is very interesting question and also very tough to answer with kind of a very direct points here. So what we do as part of one of the European Commission funded projects focusing on agricultural innovation systems, so just I would like to highlight the importance of capacity development at all levels, especially making sure that the Science Policy Society interface is happening from the beginning. So at the innovation niche partnership level, that is specifically focusing on local level, addressing some key issues, the institutional level innovation support services, agriculture support mechanisms that includes various institutions, including input suppliers, etc. And then the policy makers at the policy processes. So we try to engage them in a multi-stakeholder engagement mechanism and focusing on the functional capacities. Like functional capacities are often overlooked and not focused during all our interventions. Mostly we focus on the technical capacity development leaving behind the functional capacity development. When I say functional capacity development, it focuses on how we could navigate the complex institutional landscape at the country level. There are many different institutions, including private sector, farmer organizations, and National Agriculture Research Systems, Extension and Advisory Services. Those are called bridging institutions. So how they could navigate the complex institutional landscape, given that their reporting lines and their governance are completely different, and sometimes the mandate also varies. So they do not have any obligation to come together, but there should be a voluntary multi-stakeholder engagement mechanism. Everyone should understand that their research should be need-based and also demand-driven. And then sharing and the exchange of knowledge and information through the multi-stakeholder innovation mechanism is crucial. And this also looks at the learning part, so how they can learn from each other. And then the entire process, how this can feed into the policy processes through the policy dialogue, etc. So these are all some of the key elements, how we can ensure that multiple stakeholders can understand their importance in the broader domain of addressing several key issues, and how they can come together and develop our co-innovation and co-creation process, or co-development of technologies and good practices. So this is the type of approach we adopt, and we have been implementing several interventions at the country level. So more than 20 countries, we are focusing on strengthening the functional capacity development, which often ignored by everybody. Suppose, if we talk about the capacity development, we talk about sustainable land management or water management, soil fertility management, or input management as a standalone technical areas of focus in itself, overlooking the functional capacity that is needed to enhance the efficiency of our work. So that's the type of focus is needed, but it's not very easy. We need to identify the right entry point for this type of functional capacity development. The institution should be capable of leveraging and bringing together the various organizations in a multiple stakeholder mechanism. So this is the type of thing that we are trying to address to ensure that several stakeholders are engaged in this process.
Thank you. Lene?
Very good answer. Thank you for my panelists online. I would like to stress the FAO, where they did quite recently said microbiome is a missing link between food, nutrition, and health. That was immediately making us relevant for, feeding the world, nutrition, livelihood, etc. The next part was then immediately after that, what happened was we had the UN Food Summit. Nothing was mentioned about microbiome. Nothing was mentioned about relevant new research, which had paradigm shifts when we have possibilities of really making available, affordable, accessible biomass into growth, gut health promoting food products, and also enabling that we don't need so much pesticide. So I think what we need to do, and we have the area and the experts and the expertise in the whole microbiome community, we need to make ourselves relevant for the crisis. The poly crisis, it is important for climate, it's important for, biodiversity, pesticide use, other types of plant protection, the soil health to sustain. It is for being ready for the next pandemic, you need a more robust public health. So, and we have all that in our area of study, and we are not being taken in because it's too complex. So we need to be able to communicate in this kind of, not one-liner, that's not possible, but a little more, and then make ourselves available, but also express, communicate, so people understand that it's relevant for the situation we are in, and it's urgent.
Thank you. Thank you so much, both, because I think this is exactly what you emphasize, entry points and the way how we communicate about excellent work that we are doing. I'm really enjoying this discussion. I hope the audience in the room is. Are there any questions from the audience to the panelists? No? Okay, I will continue if anyone wants. So one of the issues that was also mentioned at the beginning was the regulatory landscape and how do we make this transition. So our last speaker also said we make to ensure the robustness, the efficiency. I fully agree on that. But I think we also know that the regulatory landscape is not really ready for the kind of products that we are working or applications like the nice example of the SynComs, etc. Because a lot of regulatory was just taken over from the old chemical ones. So one, perhaps a challenging question, but if you could advise the regulators how to change it, what would be like one or two key issues that you say should be considered or changed in the current regulations to enable better and more efficient registration of microbiome-based products? Difficult one.
I think we can make it comparatively short. As I told you earlier in the presentation, you have to distinguish between a crop protection agent and a biostimulant. There is a new rule for the biostimulants, which includes PGPMs, and for those, the regulations are comparatively lax or easy. That means we don't have these big problems to bring a product on the market if you can show that it has not an effect against any phytopathogen. If this is the case, then you get all the problems that you have with crop protection agents. If there is an effect against, for example, phytopathogenic fungi, that means there needs to be some compounds produced by this organism which may have negative effects on humans as well. Just think about all the antibiotics which can come from microorganisms. And up from this moment, you are really in this area to have a lot of studies to be done. And for many this is so cost intensive that it will not be done. So let's stay in this biostimulant area where the admission is much easier.
Okay, we have some answers on this side.
I would actually argue the contrary. I think that this division is very problematic. And I think it is because if you are thinking about, if we are staying in the plant area, a biopesticide, it could be a biofungicide and it could work just by outcompete the fungi. So it does not need to produce any toxic compound. It may just be better at colonizing. So by grouping all these, so I think actually there will be quite a lot, and in many cases also, if you have something which are stimulating the health of the plant, it may actually also be more robust against pathogens. So there's a side effect there. So I think that it's an artificial distinction between something which are promoting the growth and which is something which are antagonistic against something. And I mean, we had the same debate years ago about growth-promoting bacteria that you were giving to pigs, and so now growth-promoting substances, which was antibiotics. But you were doing it for growth promotion because then you are allowed to do it, whereas if you are actually doing it to kill pathogenic bacteria, which is what you were, but you are not saying, then you are not allowed to do it. And I think we should not get the same problem here, that when you are trying to put something on the market with a false label just to get into other regulations, I think we should argue rather that these things can be tested and they should not need to be over-tested, that there is a lot of natural variation happening, winter, summer, drought, non-drought, so populations will change. And if we add something and you do not see changes which are significant, different more than those, in my opinion, there's nothing to worry about. You cannot stop summer and winter. So if you're just within those kind of natural changes you're seeing in the community, I think rather we should argue for less strict regulation and focus on the specific. I mean, there are specific issues to worry about, that you don't kill beneficial fungi, mycorrhiza, and that kind of thing. So I mean, it is very easy to define what to look for, and rather do that.
Briefly,
I would like to continue from what you are saying, Søren, because I think the most clever would be if we could identify the ones which are the safest, the easiest, the nature-based solution where you are strengthening something, out-competing the other ones. And I think that if we could try to do what EPA did in the 80s in US, fast track the most easy, the ones where we can see there are no problems in. And then we will get people then in regulatory understanding a bit more about prebiotics and probiotics where we have no problems. Right now they are stopping, and they have been told, the companies who have pre and probiotics ready are being told that it will not be even starting to be looked at in regulatory between more than 2 to 3 to 4 years from now. I mean, it is really serious. So I think we should be very good in selecting the easiest, the low-hanging fruit, and then to go and make the sense of urgency and have a fast-track evaluation. Because it's important for the world, the health, everything.
Thank you. I'm over here.
Just shortly to amend, if you have this outcompeting effect, which is not a direct killing effect of the pathogen, according to my understanding, this would not be a crop protection agent. That would be a biostimulant. And then all this long list of tests, ecotoxicity, human toxicity, all this very cost-intensive analysis would not be required. So that would be an advantage.
Okay. We also have one comment from the audience. So.
I agree with Søren, because I think that it's impossible to discriminate between plant growth promoting agents and biocontrol agents. Also in SIMBA microbial consortia, we have strains that have multiple function. And I'm wondering regarding the regulation, because in human, when we perform transplant of our gut microbiome, we did not have so strict regulation. We apply transplant, fecal transplant, to improve the health of some people with dysbiosis or Crohn's disease. If we think that also in agriculture we can perform transplant of soil microbiome, a complex microbial community, is impossible to discriminate between the different functions of microbiome. So we have biocontrol agents, but also PGP direct activity. So in my view, it's impossible to schedule our consortia or SYNCOMs in one or two categories. So it's strictly to consider. Suggestion for the policy to consider the complexity of this microbiome and to see the entire effect of on plant productivity, on plant health, as we are doing in the human health.
Thank you. I think we are back to square one. It's a complex, and we need to make people understand how complex it is and what are the best solutions. Being fully aware that we are slightly over time, I would just like to end this round with one question to all of you. I challenge you to give one sentence, one take-home message that you would give to a young scientist considering to start to work into the microbiome research field. So what would be like your vision, your take-home message, your motivation message for someone who is now thinking, should I go there or not? Let's start with you and then...
I think Lene has already said it. I think it's the answer to all the major challenges that society are facing.
Thank you.
Well, I would be a little bit more specific and say, well, from a bioinformatics perspective, again, I think there are so many unsolved challenges that there is enough work to do for like, I don't know, many PhDs.
I think I would say to a talented young student, I would say, choose it because it is difficult and it's going to be damned important.
Thank you. Anton. Okay.
I can make it short. Don't give up easily, but stay critical.
Okay.
It's really cool.
It's really great. So, and last but not least, our online panelists. So what would be your message?
Yeah. Thank you, moderator. So I would suggest that to think about the potential for scaling up and think about the potential for applications at the producer level. And this is critical. Sometimes there are a lot of enthusiasm in the basic research, but sometimes, of course, it is needed. But at the same time, we need to think about the future, how we are going to apply and who are our clients and how we can engage them from the beginning.
Thank you. Thank you for these nice closing words. Thank you to all of the panelists. You were great. Thanks for the interesting discussions. And on this positive note, I would like to close this panel. Thank you for your attention and looking forward to the next section. Thank you all.